Bueno, muchas gracias por recibirnos aquí. Es un placer muy grande poder compartir con todos ustedes, aprender también, partilhar esta experiencia. Y ahora voy a mudar para inglés. Yes, again, thank you very much for Ocean Blue 3 and Pelagika Kunja for making this happen. And it's a great pleasure. We've been very excited to come here for since last year. So we finally made it. So I'm very happy about that. And so with that, I'll start by a bit of context. By now, I think most of you, I've explained to most of you where, if you didn't know already, where the Azores is. So essentially, we're kind of halfway between Portugal and the US, a little bit closer to Portugal, but on the top of the mid-Atlantic ridge on the North Atlantic. So this is it's not dry as Cabo. It's very lush. It rains a lot. It's very luxurious. And there's a huge diversity, especially of megafauna. There's a lot of different cetaceans, sharks, rays, turtles. So it's it's a great natural history lab for these sorts of animals to understand these sorts of the ecology and the migrations of these habitat use, etc. So this is just an example of a paper we published a few years ago with, of course, this is a bit not completely a correct picture because it looks like all the animals are going away from the Azores, but of course they had to come to the Azores before we tag them. And this is examples of work done by our colleagues that do cetacean work. This is fin whales that come across the Azores on their migration north to their feeding grounds of Newfoundland and Greenland. These are tunas that we've tagged in the Azores, big-eye tunas. They go, they basically use the whole North Atlantic. Each animal is doing its own thing. These are blue sharks, essentially, that we've tagged in the Azores, and this data set has been used thoroughly in different research to address different research questions. And these are birds mostly caught in shearwater. See, they go all the way from the Azores where they nest during the summer, all the way down as far as South Africa. So this is quite amazing. So the Azores is kind of in this Atlantic crossroad in the middle of the ocean for many of these different taxa. And of course, this is not new for none of us here. We're all interested in understanding things like where the animals are going, why they are going there, how are they getting there, how they can get there, how they navigate, how they use the energy, etc. And so this is part of our motivation. In addition to that, we had a few years ago to address the issue of related to ecotourism and the impact it could have or might have on the animals it targets. The two main species that are the focus of, or the main large pelagic species that are the focus of ecotourism in the Azores are the blue shark and the Chilean devil ray or the sickle-fin devil ray, Mobola terrapacana. And so, especially regarding the sharks, people were concerned that because they're attracted with chum, people were concerned now if you're attracting sharks with chum and putting people in the water, they will associate people with food. Is that a risk for people? Will they become more site attached or resident closer to the islands and eventually start using the coastal areas where the people in conflict with people uses or human uses? And so there's an issue of risk management. And also the fishermen were saying, well, now you're attracting sharks here, they're going to compete for the same resources as us because they're going to become resident, they need all the fish. And so, but it was a discussion that no one really had any data to support their points of view. And of course, there's fundamental ecology questions, and all of this wrapped up is essential to support smart management decisions. But the traditional methods we had available at the time didn't really apply or weren't ideal to answer these sorts of questions. So if you want to understand why a mobula is aggregating after thousands of kilometers migrations, why are they aggregating this specific seamount? If you go there and you restrain it or you tag it invasively, it might leave and you don't know if it left because it wanted to leave or because it's responding. The same with sharks. If you want to understand if a shark that is attracted here will become more resident or site attached. If you do this to the shark, it's likely that the shark will go away. Does that tell you that the shark is not being impacted by the chum, or does it mean that it doesn't like to be handled like this? So we had to come up with ideas, and one of the things that at the time was available was the Criticam with a suction cup for the Mobulus. But not only it does not work very well for the Mobulus that it was tested, I think essentially Alfredi, and also I think some oceanic mantas. But the Tarapacanas, they have even more mucus. They have a thick layer of mucus that gives that greenish, that olive color is mostly mucus. So if you remove the mucus, the color is different underneath. And so we tried a couple of times with something similar. It's just ridiculous. They basically just slide on the back and they would just go like this, and then after 10 seconds the tag is out. So no way we could do that. So one day we were diving on a seamount where the Mobulus aggregate, and we saw a Mobula just got stuck on an anchor line. A lot of you have seen this, and they keep going forward. They don't have reverse. Can't do reverse. And so the idea occurred that maybe we could use this to our advantage if we have some sort of harness, some sort of way to use this shape with a protruding head and these large shoulders and fins, and some sort of sensors or tag trail behind. Maybe it just stays there and we can do it non-invasively and then not impact the behavior of the animals and understand why they are there in the first place. And so the first time, this is really the first time that it was done. It was in an expedition. We were lucky to have a cameraman, and at the second or third trial, I managed to lasso one Chilean devil ray. You can see the tag is nothing like the tags we brought here. It's just a piece of synthetic foam that we carved in the shop and put a mini pad attached to it and a radio transmitter like this one, just to prove the concept. And the concept worked. And then some of you know Fred Boyle, who lives in our island, and he said, well, it can be done for sharks as well, I'm sure. And I told him, I believe you, but I want to see first. So he actually did it. This is Fred Boyle. This is the first time there was tried on a shark. And he lassoed the big blue shark, and he was fine. So after that, we started doing it. And now we do it all the time, and it's fine. And then we went from this very basic thing. It had an acoustic transmitter, a radio, and an old non-functional splash tag that never really produced any data. It never really worked. We went from this to what you see now. But the thing that has not changed much is this very simple thing, which is the harness. We started with some nylon and a tube, so it doesn't hurt the animal. It doesn't dig into the flesh. But the most important thing is the galvanic link, of course, because it releases the harness. And the nice thing is it never fails, because there's no electronics, there's nothing to fail. It just dissolves and it eventually opens. It can take a little bit longer, a little bit faster, but it will dissolve. And a piece of tube from the bicycle tire, or the tube actually, to work as a stopper. Because in the beginning, when the sharks were going for long or sharp turns and things like that, the harness would tend to open, and then it would release. And then we came up with this very, very simple thing, which is just a piece of old rubber, and it's a stopper that allows us to adjust it to the girth of the shark, and it stays in that size that we want. And so it keeps it from shedding the tag prematurely. And so we did some initial trials to understand whether or not this technique had the potential to affect the behavior. So we compared essentially the vertical behavior of the animals that we tag immediately post-tagging, compare that with long-term tagging data that we have, and we didn't see any significant differences. So we were happy with it. But then of course, we had a very, very long wish list that we presented to our colleagues from the engineering department that we collaborate with from SEA. And of course, you can see here, basic things like pressure and temperature. Okay, it's not very difficult. But then, okay, GPS makes it a little bit more difficult. But wildlife Wildlife has some sensors that have that. Accelerometer, other manufacturers have that. Magnetometers, gyro, speed meters. But there was nothing in the market that combined all these things. So we proposed this idea of having this platform that can be sort of customized for the sensors that we want to combine and have these concurrent sensors and readings for the same deployment, for the same animal. We collect all these variables. And then, of course, it had to be pressure resistant to 2,000 meters because these guys dive super deep. We know that from previous studies. And so this is a big challenge because all of these things, you know, the 200 kilos of pressure in 1 square centimeter is just unbelievable. It's a lot. It's a lot of pressure. But the guys from that collaborate with us from SEIA, from Portugal in the north, they're very smart, they're very clever, they have fancy tools, and they put their heads to work. And after 4 versions, we ended up with this camera system and the multi-sensor tag. It came a little bit earlier, but this was the current version of it. And of course, it needed a lot of, to get here, a lot of work of validation, a lot of frustration, a lot of things that go wrong, a lot of expeditions, especially abroad, when you have an 8-hour difference and something is not working and you're trying to get the engineers on the phone and explain you how to open the tag, how to weld the wire that has broken, or, you know, yeah, it's a long path. But here we are, fortunately. And this is an animation, supposed to be. Okay. It froze for some reason. Let me. Oh, Jesus. Okay, it's going. Okay, this is a nice animation just to show the concept of this customized thing that combines sensors from ATS, sensors from wildlife, custom-made sensors, cameras, etc. In one package, in one full package that allows you to have all this wealth of data when you deploy the tags. Okay, so in summary, basically what you have here is the main body, basically, which is the titanium housing that holds all the electronics, the batteries, the cameras, the sensors, the MIUs, the temperature sensors, etc. And the red LEDs. Why are they red? Because most of the animals that live in the deep sea, they can't see red very well, or they can't see red at all because it's useless, because it doesn't penetrate that deep. And so we use that not to disturb the animals or the prey that they are targeting. This is very, very smart system, which basically is a paddle wheel. It's passive. It just, the water makes it, going through it makes it spin. And because there are alternating magnets, the magnetometer in here will measure the frequency of rotation, which we know corresponds to a certain speed, which is really nice because it's super simple. I like simple stuff. And of course, we need the assets to recover the tag once we deploy them. First, we get a general position from the Argus transmitter, yes. So it's a spot tag. And then when we're closer, then we can zoom in using the radio with a directional antenna. Okay, it's a bit more detail. I'm going to talk to you, also present the multi-sensor tag. It's a tag, it's smaller, and it's designed for when you don't have as much need of video, but you're looking for good geolocation. So you want to know where the animal is going with great accuracy, with great detail. So what we did is, it has the same MIU in here. It's a CATS MIU, so it measures acceleration, the temperature, magnetometer, and then it has radio for recovery and spot for recovery as well. But in addition to that, on the top, you have a fast lock GPS sensor, which is a great instrument because as soon as it breaks the surface, even if it's just a second, it will collect a snapshot of the GPS constellation, and then we'll post-process that and get very accurate locations. For example, the Argus can give you a large error depending on the quality, but it takes a lot of time to send messages to the satellites. Basically, it uses the Doppler effect, which means that is not very accurate. And depending on the number of satellites, it can be quite like hundreds of kilometers of so that's why sometimes you get a shark in the middle of Africa and not in the middle of the Atlantic Ocean. This one also has the paddle wheel. So, but the thing is, there's a lot of people using accelerometry out there before us, and the difference is that it's attached to the body of the animal. So everything that the animal does is related to the tag, or the tag is reflects that, or the sensors in the tag reflect that movement. But in the case of our tags, they're being towed, so they're not rigidly attached to the animal. So is it useful? So that's why we did a lot of work to validate this. So this is an example from a Chilean devil ray. It's climbing from a deep dive. And I'm going to pause this for a second. No, maybe not. Yeah. Okay. Just to explain these graphs, because I'm going to show a couple of them. So basically this is depth, it's climbing. And here, this is the frequency of the wave of the wing beats. Okay, this is accelerometry. So basically one cycle of 4 seconds means takes 4 seconds from going from here down and up again. So it's a full cycle. In this case it's 4 seconds is this wave. And here the colors represent the amplitude. So the amplitude is quite constant, and if we have differences in color, it means that light colors you would be doing this, warm colors you would be going like whoa, woof, woof, like when they want to speed up for some reason, okay? And so of course we used the video. You use the video to make sure that what we see is what's happening. And you can see here the wings, you see the line, they correspond exactly to that wave. It's a bit noisier, it takes more time to process the data, to clean the data, but they show the same thing. This is an example from a reef manta that we tagged in Hawaii a few years ago. Like the one from today, it only had, because they have the same behavior, it happened the same thing. It only had the harness over one cephalic fin. And basically what you see here, when she takes off, you see a lot of energy here. You can see by the paused image that she's going like, oh, I'm going away. And you can see also here the amplitude also is reflected there, and then it decreases. See, this is about 30 seconds later. So she takes off, she's kind of spooked like today, and then you can see on the footage, she will start to slow down after 25, 30 seconds, and then she will just be super calm. So this gave us confidence that it works well for mobulids, even though we're not measuring the traditional sway that people measure in sharks. We're actually measuring this. So we're not using the same axis. We're not using the sway. We're using the surge. Thank you. Yeah, you see, she was going at 2 seconds. She was going really fast. Now she's so slow that you can't even see the mobula because the camera floats higher when you don't have speed. When you don't have speed going through the water. See? So she's kind of just gliding. You see barely any movement of the fins here. Okay, I'm going to go to the blue shark. This is a blue shark going on a dive. And then you can see here the frequency is about 4 seconds. This is in hertz, so you have to invert it. And then it levels down and it starts to climb. And you can see a change. Look, and you can see that change in the head. He's going slow, and then when he starts to climb, he's going like this. Faster, but lower amplitude. So we see that in the data, we see that in the video. That gives us, again, confidence that we're doing a good job and the tag is showing what the animal is actually doing. This is a whale shark. It's very interesting because these are massive whale sharks. We use a clamp on the pectoral fin, and we see the same thing. We see these long 8-second cycles. They're really slow when they're just, we're not going for the food or something. They go, it takes 8 seconds from the tail to go from one side to the other and back. And you can see that on the head like the blue shark, except it's much, much slower. We also took the opportunity when we were in Hawaii with Carl Meyer, and we had the access to tiger sharks that they were tagging. So what we did is they put a fixed accelerometer in the pectoral fin, like they always do, and we put the harness and the toe tag, and we compared both, and we were happy with, again, ours is a bit noisier, but it shows the same trends in behavior. Basically, when you see one is changing in one direction, ours is also changing in the same general direction. And so, but we were a bit also concerned with, okay, they can relatively well show the movements or represent the movements and the behavior of the animals, but does it have an impact? Of course. And you think, well, it's dragging something, it's dragging something. In the case of a bird, it's very important to not add too much weight, because then it will be tired faster, it would not be able to do the things they normally do. For the marine animals, weight doesn't matter. What matters is the drag, because they have to swim through the water, and that's what they use energy for most of the time, is to move themselves, propel themselves through the water, because water is very viscous. And so what we did was we did computer fluid dynamic models for the animals and for the tag. So we estimated the total drag of the animal for different sizes and compare that with the tag. And so what we wanted to is to determine the threshold of 5%. We didn't want to add more than 5% drag to the animals. So with this, and we're being conservative because we just estimated the drag for the animal in the stiff position, let's call it like that. But we know that when the animal is actually moving or swimming like this, the drag will increase by up to 3-fold. Okay, so we're being quite conservative. So what we see for manta rays or devil rays in this case is that even for the camera tag, which is the largest one, anything above 2.5-3 meters is acceptable to tag with this tag. Sharks have to be a little bit bigger because they're slender, more slender, but this one, the multi-sensor tag, is adequate for most of the animals that we've been tagging, and we continue to tag above 2.5-meter sharks, for example. Okay, why is this different from the other one? This is the other drag, and this is the, okay, it's the same. I forgot to delete the other one because these were more recent fluid dynamic models. By the way, we did these new ones because we were studying the drag added by the remora, and what we learned is that the remora actually add more drag than our tags. These are big remora, like the same size, approximately the same size that we see here. Of course, these, in relative terms, these mantas are larger, so the added drag is less in percentage. But for these guys, it can be up to 5%. A couple of or remora. Sometimes they have more, and you see that in whale sharks as well. You know, we've seen. I'm sure most of you have seen whale sharks carrying 20 remora in their tail, which probably is even more expensive energetically, because the tail is what is the propeller, and if your propeller is in bad shape, then that probably has a larger impact. So the point is, these animals, they have to deal evolutionarily, they have to deal with this, with added drag, and this is probably throughout their lives, and we're talking about deploying up to 24, 48 hours. That's usually what the length of the deployments for the larger tag. This other tag, the multi-sensor tag that we use to collect accurate locations, depending on the animal and its behavior, we can collect up to, if the animal is surface oriented, we can collect up to almost 28 fixes per hour that it spends within 0 and 1 meters of depth for the mobulas. For the blue sharks, we got up to 29 locations per hour that it spends within 0 and 1 meter. So that's pretty interesting. That's pretty useful when you want to understand, for example, if an MPA has an impact for a certain animal, if depending on your question, if you need high spatial resolution, which habitats are more important? Is there a critical habitat here that they use more and more frequently? So this is very useful for those sorts of questions. And here's what should happen when everything goes well, which was almost the case today. It was not like, and you can see that also the difference when you're free diving, you can be a little bit faster and slicker. And also the big difference is the head of the Tarapacan is not as wide. It's much easier to abrazar. But again, we are only showing the nice parts. Of course, there are many days that we spend the whole day in the water and waiting for the situation and waiting and waiting and waiting. And sometimes we do a lot of work in 15 minutes. Sometimes in 15 hours we get nothing done. It's the animals that, and these are some highlights that I wanted to show. This is how usually when the camera is the leader is coming from the top of the head, which is usually what we aim for. This is the kind of view that we have for the mobulus, especially, and it's great for interaction with other conspecifics, other species, etc. This is kind of, the screen is a bit, but this is, you can see this is great also to detect. This is like 150 meters deep. This is around the Princess Alice Seamount. This is also great, for example, to use these animals as platforms to map habitat around the areas that they like, and also to understand which habitats they prefer, which habitats they use. And we have even deeper than this, but of course, as it gets deeper, there's less light, and then the lights don't go that far. You can see 1 meter, 2 meters in front of the camera, not much more than that. But here you can see there's other conspecifics. There was a apparently pregnant female also swimming above it. So I'm going to show something about this. So this is what we think was the first time that we filmed a potentially pregnant female, because we see what looks like a pup kicking on the left side, which is the functional uterus of the mobulus. And so we can prove this, obviously, but it suggests that, well, they are big, they are very fat, some of them, and then if you see some movement that could be a pup, then it suggests that you should be looking in more detail, maybe try to collect some blood samples, for example, which is not easy, but try to understand if this area is important or not for parturition, potentially. We have never seen juveniles. This is a very interesting case. We found this behavior we call the power dive. And these animals that we thought were very chilled, just slowly swimming at the surface. Well, first we knew a few years ago they can go almost to 2,000 meters. And now we know they can go super fast when they go deep. And what you saw was not the animal spinning, was the camera spinning. Because what happens is when the camera, when the tag goes faster than 10 knots, like an airplane going up, the fins, they stall, and so they are no longer efficient to keep it stable, and so it spins. But as soon as it slows down, then it stabilizes again. And these power dives are amazing, because then we look at the data, and what did we see? Remember what these lines are showing. So here you have depth. So it's going from 0 to 800 meters. Okay, so it's just chilling at the surface. This is just noise from the waves, the tag at the surface just bouncing back and forth. And here, so you have all this noise. And then it decides to go down on a power dive. And as it starts to descend, you see all this energy. And basically what it means, it went like super crazy fast and high amplitude, lots of energy. But halfway through the descent, there's nothing. So it's basically gliding. So it keeps the same slope, meaning it's going the same speed, the same vertical speed. So it's going super fast, not using any energy. And of course, this is a puzzling behavior, because they're negatively buoyant. They could just go down, no effort. But we think it could be related to a thermoregulation strategy when they're deep diving in cold waters. So it's possible that they may be doing this high energy movement to store heat in the muscles, and then bring this heat to the brain through this rete mirabilis they have, which had been described earlier as a mechanism to lose heat. We think it can be used to lose heat in very warm water, but it may also be used to warm the brain in really cold water, because these guys can dive to, we have dives that almost 2,000 meters. It's 3 degrees Celsius. It's freezing cold. And also, when you start to look at the three-dimensional behavior, the things that you see like a line going like this, it's a dive, it's a V-shaped dive, it's a dive. No, it's not like this. They're not bidimensional, they're three-dimensional, or they're four-dimensional. So this is a section of climbing section from a dive, and they're not going like this like we see in the 2D. They're going like this, like a corkscrew. They're probably sensing the environment. They're looking for prey potentially. We don't know exactly why they do this, but certainly is more complex than we anticipated. And this is interesting examples of things that we can see. This is a blue shark that found a carcass of a dead sperm whale floating and is going for a free bite, free lunch, which is something hard to find in the blue open ocean. This is the same animal found also some tourists, so he's doing some people watching. So the roles were inverted. They were trying to do shark watching, but they were surprised by the shark watching them. And this is the moment where we prove that the whale sharks in the Azores, they actually feed on fish. They don't feed on plankton. You see there's a bait ball, and they had a camera on it. And they in the Azores, the whale sharks are usually accompanied by schools of tuna. And the reason for that is because the tunas will herd the fish into tight bait balls, and then the whale shark can go in and they can eat the bait ball, because otherwise they go to the bait ball goes in its back, is like a big hair on the really big hair on the shark, and nothing happens because they can't prey if they don't have the help of the tunas. And this is also a camera shark. We were trying to shoot film sharks from the sub, and he also the same thing. He came and he filmed us when we were trying to film him. It was a very, but the story behind this is even more interesting, because we were looking for the sharks from the sub, and it's not easy because the sub is slow and you don't have much visibility and all of that. And one day before, we had realized that after a storm, most of the tuna left, but some whale sharks stayed. And they stayed, and they all went to this seamount MPA, where there's a big resident schools of jacks and barracudas and bluefish. So all pelagic predators. And so the whale sharks started to show up only there. And we realized after a while they were trying to recruit these fish to replace the tunas, because they would come in, and this is what happened here. So we were there with the sub, and all the jacks were around the sub. So they adopted the sub as their aggregation device. And the whale shark comes in, not to check us out. He comes in, he parks next to the sub. All the jacks jump on the whale shark, and he leaves with all the jacks. So this was quite exciting. And then we've used this also to bring to light some exciting new behaviors of the mako sharks in the Azores using this tag exactly. So we can measure speed, tail beats. We can see the differences between night and day. These speed versus the red dots over there. This is also very interesting. You can step on unexpected things, like what you see here is a whale shark. This is the bottom at 1,450 meters, and that's a common remora over there. The same that we see here. That's a common remora. We had no idea that the hitchhikers, well, we speculated, well, do they deep dive because they want to get rid, they want to evict these parasites, or these hitchhikers because they are producing drag? And we had a paper published not very long ago that also explored that hypothesis, that was looking at the hypothesis to explain deep diving of these pelagic animals. But in fact, they don't care. They go. And it's not just the remoras. On this side, you'll recognize a pilot fish. the same dive, we didn't see so the pilot fish is not attached, so it has to swim, it has to follow its host. So it's quite amazing that it can go through 900 meters is when we see it, but I'm sure it went all the way. Oh, well, I think he went all the way like the Remora, because we also see him on the way up. So likely the pilot fish also was able to do 1500 meters, and you know, temperature is 3.6 degrees down there. The pressure is overwhelming, and they have to go through hypoxia around 800 meters. So they can. It's very, very interesting. So that also allowed us to start a new research line. So we took advantage of having the Ocean X ship last year. We collected some remora. We implanted heart rate and internal temperature sensors, and then we simulated the same dives, and we found they're amazing animals. They can reduce their heart rate when they're down deep in the cold to extreme bradycardia, to 6 beats per minute. But when they're recovering, they can go to tachycardias of 120 beats per minute. And they're amazing. And one of the things that we found, and then they can heat faster than they lose heat. And they're not endothermic animals, but they have some strategy. And they can, you know, they can function with very small difference between external freezing temperature and very cold internal temperature. And one of the things that we observed also by accident, once we finished the experiment, we sacrificed the animal to collect tissue, to run biochemistry, and the heart was one of the tissues that we had to collect. And we had the heart beating outside of the animal in a Petri dish for almost 4 hours alone. And we would cool it, it would go into bradycardia, it would slow down super slow, we'd take it off the ice, would start beating fast again. We cooled it, we couldn't believe what we were seeing. So I'm still not sure what to make of it. And so, of course, all of this is all interesting, but how can you use this to improve management and conservation? So this is an example of a paper that I should be writing right now. I have all the data ready. It's about the shark diving in the Azores. So this is the main location, is between the two islands in the north slope of the channel. This is a seamount MPA. This is an alternative site these days because this is cheaper, it's closer, it's faster to get there, and there's lots of sharks as well. And so basically what we wanted to see whether or not the sharks stay. Do they become habituated? Do they become resident? And keep in mind, these are blue sharks. They use the whole Atlantic. They will use the whole North Atlantic. You see, remember the graph from the beginning? They're not sedentary animals. They're not territorial. Their range is super wide ranging. But what we see is that the longer time elapses since we take the chum out of the water, typically the further away we're going to find the sharks using the fast log GPS. So that tells us that no, the sharks, they're there because there's food, and the chum helps you to attract the sharks and stay on the boat with the divers, because if there's no sharks, you can drop 10 tons of tuna, because if there's no sharks, there will be no sharks. The same thing we did for the mobulas, to understand whether or not the impact of having all these boats and all these divers on a daily basis or almost daily basis, the weather keeps the boats out some of the days, so they can dive all the days. What we see is that, well, we kind of see two different things. Some animals stay longer, up to a week. Some animals just take off. So what we think is these animals are sort of rotating. There's one specific seamount here where we did this study, Princess Alice. There's other seamounts where we also have detected them with acoustic tags before, and maybe also going to other islands on the eastern group where there's another hotspot. So it tells us that possibly the effect is being diluted among animals. So where are we going to next? Of course, we continue to have all these ideas. Some are good, some are worthless probably. But so we're working on testing now a new oxygen tag on this Nautilus project. Basically, it's a tag that is able to, it does all the accelerometry, depth, temperature, and it has a dissolved oxygen sensor to try to understand how these animals go through these different landscapes of dissolved oxygen, and potentially how the deoxygenation of the oceans might impact their ecology as climate change and deoxygenation is advancing. So this is another tag that we tested last year, and it did some improvements to use now. It's a tag designed to measure internal temperature, to test that hypothesis of whether or not they're using, well, one, whether or not they're using that behavior to generate heat before the deep dives, and also how they use their thermal landscape, and also understand how the warming ocean might impact the ecology of these animals. This is a bit more invasive. It has suction cups, but it also has a spike that has the temperature sensor at the end. The nice thing about it is we think that if you understand what's happening with 2 or 3 animals, then you can probably extrapolate that to the population, because the physiology shouldn't be that different. They're all adults, at least here where we work in the Azores, they're all adults. They're males and females, might be some difference, so it'd be ideal to tag a couple of males and a couple of females. Hopefully we'll get this done as soon as we get back for the rest of the season. Sorry. Also an idea, it's just an idea on our head for now, is use the similar methodology to measure heart rate of the mobulus. So having some sort of sensor like the ones most of us have on our watches, light-based, that can penetrate the muscle, and with that get information about the heart rate of sharks and mobulus, because we know very, very little about the physiology, the heart physiology of these animals. And the other tag that we hope to, we have some proposals submitted, this is a very interesting idea, is to use this sort of approach and have also a micro sonar tag to address the distribution and diversity of the mesopelagic prey, because all these guys are diving super deep into the mesopelagic. We think they're using the mesopelagic for food, but we have to show that. Plus, we don't know much about the mesopelagic, and there are a lot of people these days that suggest or defend that we should be extracting mesopelagic biomass for food, for fish food, for whatever, for protein, and we still haven't fully understood how important this mesopelagic prey, this biomass, is to these a lot of these megafauna, including mammals, including sharks, rays, etc. And so this tag that we are planning, we're kind of already starting to develop it, but we still need a bit of funding to push it further with collaborations with very smarter people than us. The idea is to have the micro sonar, which already exists, integrated into a camera that with a microchip that or microcomputer that basically, when detecting a specific acoustic target, it will trigger the camera. So you can actually have an image of what is the target that the acoustic sensor is seeing. And hopefully, if you get a sequence, maybe you get a feeding event, or you get at least refined information to feed into the models that try to predict the distribution of mesopelagic prey. And so with that, I'd like to thank you all for your attention. I'm not sure if it was the 45 or the 15 minutes. I lost track of time, but I hope you enjoyed. Thank you very much.
Thank you.
And questions, if you have any.